Welcome to our webinar series, Laboratory Good Practices from Greiner Bio1. My name is Ruben Daum and I'm Product Manager for our Cell Culture Labware. In the third part of our webinar series, I'm talking about selection of surface treatments in cell culture. If there are any questions, we provide our contact information at the end of this presentation.
What will be the content of this webinar? In the first part, we will take a look at the different cell types and their requirements, followed by addressing the different cell culture surface treatments that are required for successful cell culture of these cells. Next, we'll look at a few applications to get an impression of what the different surfaces are suitable for. And finally, to sum up this webinar, an overview of all the cell culture surface treatments is provided, which helps with selecting the appropriate surface. So let's start with the first part, cell culture surface treatments. Our human body consists of around 100 trillion cells. Everything starts with one stem cell, which differentiates into all these different cell types, as we can see here on the left-hand side. And these different cells, they form the organs which fulfill all the different functions in our body. Just as each organ has different functions, the cells which form these organs, of course, also have different functions, phenotypes, and properties. And due to this, they also have different requirements. And this is very important for us to understand if we talk about culturing these cells in vitro. So cell culture can be defined as the growth of isolated cells under controlled conditions outside their natural environment. Even if the cells can be maintained under carefully controlled conditions, there are factors influencing the cell morphology, cell cycle, and differentiation status of these cultured cells. And one factor is the cell itself. So the origin, the passage number, the age of the cell, also the donor, the age of the donor, and so on. Another very important factor is the cell culture medium. So in this context, the nutrients, the pH, the osmolarity, also the supplements which are added to the medium, for example, the FBS, as we will see also later on. However, in today's webinar, we want to focus on another important factor which influences the cell culture. It's the culture substrate or surface. And we learned that every cell has different functions and therefore different requirements. And due to that, it's obvious that cells also should be cultured on different surfaces. The goal of a successful cell culture is to control the cell's function and characteristic in vitro by offering the appropriate surface. Cells cultured in vitro can be roughly divided into two groups: the free-floating or suspension cells and the anchorage-dependent cells. In vivo, free-floating cells are all the blood cells, such as the B and T lymphocytes, and they have a spherical shape, also cultured in vitro. In order to maintain their typical properties and their phenotype, they also mostly should be cultured in suspension. And this can be realized by using an untreated polystyrene surface. So this surface is quite hydrophobic due to its unpolar groups. And this hydrophobicity of the surface leads to a reduced protein absorption and prevents the adhesion of these cells. Other cells which are cultured on the surface are hybridoma cells, which are used for antibody production, and also insect cells, for example the SF9 cell line, and also quite popular is culturing adapted anchorage-dependent cell lines, for example CHO cells or HEK293 cells, which can be used for recombinant protein production. And finally, as we also will see later on, this surface can be used for culturing embryonic stem cells. The other group are anchorage-dependent cells or adherent-growing cells. And these are the majority of the cells derived from vertebrates. Culture them in vitro, they form in most cases a monolayer, which can be expected quite easily under an inverted microscope. And these cells, they can show quite different morphologies, as we can appreciate here on the right-hand side. And so on the one hand, we have fibroblast-like cells, which appear elongated and show a bi- or multipolar morphology. And on the other hand, we have epithelial-like cells, which form one or even more cell layers and appear flattened and polygonal in shape. Their morphology is also sometimes described as cobblestone morphology. In order to offer the appropriate surface for culturing these cells, it's important for us to understand the underlying mechanisms of this cell-substrate interaction. And this cell-substrate interaction can be divided into three phases. So first, when a cell comes into contact with a substrate, we have electrostatic interactions. After a few minutes, in phase 2, the cells can form integrin bonds. And again, after a few minutes to hours, these integrins can mature into focal adhesions. In more detail, in phase 1, the electrostatic interaction of the cell membrane and the surface is based on polar functional groups of the substrate. And these polar groups, they can be positively or negatively charged. For us, important to know is that in most cases, this interaction is quite weak. So for example, if cells are cultured under serum-free conditions, so without any supplemented proteins, they can be easily washed away or shaked off. So proteins play an important role for this cell-substrate interaction. And the reason herefore we see in phase 2. A much stronger interaction of the cell and the substrate is mediated by integrin bonding. However, integrin bonds only can form if the integrins have specific ligands where they can bind to. And these ligands are provided by serum proteins from the cell culture medium, which are deposited on the polymer. Also possible for some cell types is to synthesize their own extracellular matrix proteins with which they can form these integrin bonds. Also possible is to provide a pre-coated surface with proteins or peptides, which also provide the ligands for the integrins. So here we have a medium cell-substrate interaction. These integrin receptors, after a few minutes to hours, cluster together and form focal complexes, which mature then into more stable focal adhesions. And these focal adhesions, they mediate a quite strong cell-substrate interaction. So understanding the underlying process of cell adhesion can help in deciding which surface I need for my cells in my application and whether I have to add serum or maybe can left it away. Now we know which properties are important for a surface in order to successfully culture anchorage-dependent cells. So the surface has to be polar in order to allow the electrostatic interaction of the cell in the surface and also the adsorption of the proteins, which mediates the integrin bonds. The hydrophobic polystyrene surface is not ideally, so in order to generate these polar groups, it's physically treated. And by this physical treatment, also called TC treatment, the surface gets hydrophilic and negatively charged. This so-called TC surface allows the deposition of serum proteins on the surface and ensures the adhesion and proliferation of many anchorage-dependent cells, such as AGK, CHO, NIH3T3, cargo-2 cells, and so on. So this surface is ideally suitable for culturing robust and well-established cell lines. For most cell culture applications, a common TC surface treatment is sufficient. However, in some cases, an advanced surface is advisable or even necessary. The reasons herefore can be the cells. Some cells have an impaired capability to attach to a common TC surface. Examples herefore are primary cells such as neurons, oligodendrocytes, or hepatocytes. An advanced surface, for example a biological coating, can support the propagation of these cells. Also, they can prevent primary cells to become senescent or to dedifferentiate. Looking on stem cells, an advanced surface can be necessary for culturing and differentiate them. And another application on which advanced surfaces can be important is transfection. So advanced surfaces can be very helpful to support the adhesion of the transfected cells and thus increase the transfection efficiency. A few slides before we heard that protein supplement to the medium can support the cell adhesion. This is why culturing cells under restricted conditions, so without serum, can also make the use of advanced surfaces necessary. So restricted culture conditions such as a xeno-free or animal-free medium, chemically defined medium, serum-reduced medium, or even a serum-free medium reduces the cell's ability to adhere. And in this case, the advanced surface can support adherence of the cells. In certain applications, such as cell-based drug screening assays, also several washing steps are included. And here, the advanced surface can also help to avoid cell loss due to washing steps in the assays. In general, we have two options to create an advanced cell culture surface for improved cell adhesion. One option is a biological coating, as we will see later on. The other option is based on chemistry. So here, polystyrene, for example, can be chemically modified. This means that a chemical is deposited on the surface. And the benefit of this method is that depending on the chemical used, different surface properties are possible. So for example, a hydrophilic or a hydrophobic surface can be designed, or a negatively charged or positively charged surface. It's even possible to incorporate specific functional groups such as carboxyl groups or amine groups into the surface. All these surfaces are designed for culturing fastidious or sensitive anchorage-dependent cells. Also possible is the culturing under restricted culture conditions. The benefit compared to the biological coating is also that it's an animal and xeno-free surface. So here we also have minimized batch-to-batch variation. It's more cost efficient. It can be stored at room temperature and it's also sterilizable. Also interesting is depending on the chemical used, a cell repellent effect is also possible. Why could it be interesting to use a cell repellent surface? So far, we have only focused on culturing cells in a monolayer, so in a 2D cell culture. For some applications, however, the research focus is highly complex and can only be answered in a 3D model. And in order to obtain three-dimensional cell aggregates, the cell adherence of anchorage-dependent cells to the surface has to be prevented. Instead, cells should attach to each other, forming these three-dimensional aggregates. We already heard that for the culturing of leukocytes and other blood cells, we can use an untreated polystyrene surface. However, for the suspension culture of semi-adherent and adherent cell lines, the surface is not sufficient, and here we need this improved chemically modified cell repellent surface. This surface opens totally new applications. So, as already mentioned, 3D cell culture allows the formation of spheroids, of stem cell aggregates, so for investigating embryoid bodies, for example. It's possible to perform magnetic cell culturing. On this surface, it's possible to culture organoids, and it can also be used for cell cultures for protein and peptide expression. Coming back to advanced cell culture surfaces for improved cell adhesion, I already mentioned the biological coding. So for some applications, even more sophisticated surfaces are needed that mimic the cell's natural environment. And this is realized by coating the polystyrene surface with proteins from the extracellular matrix, such as fibronectin or laminin or collagen. Another possibility is the use of synthetic peptides, for example, poly-D-lysine or poly-L-lysine. And depending on the application, it's also possible to use different proteins and peptides in combination. This ECM mimicking surface provides an in vivo-like microenvironment, which allows the adhesion and proliferation of fastidious and sensitive anchorage-dependent cells, also under restricted culture conditions. Examples of these are hepatocytes and neurons, and also stem cells. Regarding stem cells, the type of coding can also support the direction of differentiation, which is also quite interesting. The cell culture plates can be manually coated with peptides or proteins. These coatings are also commercially available, and the use of commercial products saves time, as there is no lengthy manual coding process. In addition, the validated processes of the commercial type can ensure consistent quality. In the next part of this webinar, we want to have a look at possible applications with the different cell culture surfaces. In this first example, we will have a look of cells cultured under restricted conditions. In more detail, HEK293 cells were cultured without serum. There are many reasons for using serum-free culture conditions. So, first of all, serum-containing medium is poorly defined, which can lead to poor reproducibility or misinterpretation of the results. In addition, to provide a consistent defined environment for cell culture, the detection of cellular mediators is improved and the assessment of cellular functions is also more precise. However, the disadvantage is that depending on the cell type, fewer focal adhesion complexes can be formed, and this leads to a weaker cell-substrate interaction, as we heard before. To counteract this, advanced surfaces can be used. And in this experiment shown, HEK cells were cultured for 48 hours under serum-free conditions on 3 different surfaces: a common TC-treated surface, an chemically modified advanced TC surface, and a PDL coating. After 4 hours and after 48 hours, cells were examined. And what we can see is that a distinct larger number of adherent cells are grown on the advanced TC surface after 4 hours, and on advanced TC and PDL coating after 48 hours. And looking on the result of the PDL coating, we also see that cell proliferation can be affected by the cell culture surface treatment. As best practice tip, we recommend to use either PDL or PLL coated surfaces, or even a biological coating with ECM proteins such as fibronectin or laminin, because these proteins mediate endocrine-based cell adhesion. Also possible is a more cost-effective alternative by using a synthetic advanced TC surface. The transfection of cells is another application in which the appropriate cell culture surface can play an important role. During transfection, foreign DNA is introduced into an eukaryotic cell, and by doing this, functional studies can be performed investigating gene function, gene regulation, and also signaling pathways. In addition, transfected cells are used for the production of recombinant proteins or viruses. There are different methods of gene delivery developed for various types of cells and tissues. The use of synthetic delivery systems, so non-viral vectors such as liposomes for example, is often limited by a low transfection efficiency and cytotoxic side effects. In this context, an advanced cell culture surface can buffer and counteract these effects. In the experiment shown here, cells were transfected with a plasmid for GFP expression. The protein has gained widespread importance to visualize spatial and temporal patterns of gene expression, both in vitro and in vivo. And in the experiment, the effect of culturing transfected cells on different cell culture surfaces was examined regarding the transfection efficiency. And what we see is that after transfection on PDL coding and on advanced TC surface, the amount of GFP positive cells was distinctly higher compared to the common TC surface. So, giving some helpful practice tips, we recommend to use chemically based advanced TC surfaces or even biological coatings for culturing transfected cells in order to get a higher transfection efficiency. The next application shows an example for stem cell culture on different cell culture surface treatments. So culturing stem cells in vitro is highly relevant. Stem cells differentiated into a specific lineage can be used as a disease model for drug screening and toxicity testing. And this is, for example, getting more and more important in the context of personalized medicine. Other potential applications of stem cells also include cell-based therapies. The appropriate culturing in vitro is indispensable to exploit the potential of these cells. And one important aspect in this context is maintaining the pluripotency during culturing. The unique ability of stem cells to self-renew can be mediated in vitro by adding some factors to the cell culture medium, such as LIF or 2I. However, the stem cell maintenance can also be supported by the right cell culture surface treatment. And in the experiment shown, stem cells were cultured on different cell culture surfaces, on a feeder cell layer, which is the most classical approach to maintain stem cell pluripotency. In addition, a gelatin coating was used. Cells were cultured on a common TC surface, on a suspension surface, and on a chemically modified advanced TC surface. Afterwards, cells were marked with alkaline phosphatase, a pluripotency marker. And the results show a high expression of this marker in the cells on the suspension surface. So here the cells form embryoid bodies, maintaining their pluripotency. On the other surfaces, cells are attached and form the typical colonies. Interestingly, more pluripotent cells could be observed on a synthetic advanced TC compared to the gelatin coating. In summary, we recommend to use either advanced TC surfaces or even better a biological coating for culturing stem cells. If the goal is to form embryoid bodies, it's possible to use the suspension surface. Even better in this context is using the cell repellent surface. In this last example, I want to show you a comparison of 2D and 3D cell culture. We already heard that 2D cell culture mimics the in vivo conditions of physiological tissue only to a limited extent. In the natural environment, cells are arranged in a three-dimensional tissue consisting of cells and extracellular matrix. A 3D cell culture has the potential to mimic these natural conditions more closely. So, for example, with regard to drug development, the employment of a spheroid culture is regarded as an improved approach to develop predictive in vitro screening assays. In addition, 3D cell cultures enable the expression of extracellular matrix components, as well as the formation of cell-cell and cell matrix interactions, which in turn is important for replicating in vivo cell differentiation, proliferation, and function in vitro. In the experiment shown here, a 3D cell culture formed by magnetic cell levitation on the cell repellent surface was compared to a 2D cell culture and a human tumor xenograft. The marker N-cadherin, a transmembrane protein mediating cell-cell interaction, was evaluated. The 2D cell culture on the right side show N-cadherin scattered in the cytoplasm and nucleus, but absent from the membrane, whereas the three-dimensional levitated cells in the middle express N-cadherin in the membrane, the cytoplasm, and cell junctions, similar to the human tumor xenograft on the left-hand side. This result suggests that a 3D model can recapitulate some in vivo-like properties. So to give some practice tips for 3D cell culture, we recommend to use the cell-repellent surface. The gravitational spheroid formation is facilitated in round bottom plates, and these plates are also ideal for the formation of embryoid bodies. To sum up this webinar, I want to give an overview of the different cell culture surfaces we talked about, their features, and also cells which can be cultured on these surfaces. This overview shows all the different cell culture treatments I talked about, and it might help to select the appropriate surface for the different applications in terms of cell adhesion. So if cell adhesion should be prevented, the CellStar cell repellent surface is the ideal choice. It allows the culturing of semi-adherent and adherent cells in suspension. And by doing this, 3D cell culture can be performed. Reduced cell adhesion is provided by the CellStar suspension culture surface. This hydrophobic surface allows culturing non-adherent cells such as leukocytes and also adapted anchorage-dependent cells in suspension. The most common and widely used cell culture is the TC treatment. Providing a regular cell adhesion, the CellStar TC surface is hydrophilic and negatively charged. This surface allows the routine culture of almost all anchorage-dependent cell lines. And finally, an improved cell adhesion can be realized with two surfaces. The chemical-based option is the advanced CC surface, which offers an increased amount of functional groups. The biologically based variant is the cell coat surface. Here we have a supply of extracellular matrix proteins or peptides. Both surfaces are ideal for culturing fastidious and sensitive cells such as primary cells and stem cells. For culturing induced pluripotent stem cells or neurons, we recommend to use the cell coat surface. Applications for this improved cell adhesion is culturing under restricted growth conditions, transfection, propagation of primary cells, and also support of directed stem cell differentiation. Last but not least, the cell coat surface is also ideal for other fastidious applications. For further information, you can refer to following literature. On the left-hand side, a publication of Sousa et al. Talks about 3D culture based on magnetic cell levitation. On the right-hand side, two application notes look deeper into applications for advanced TC and improved cultivation and differentiation of embryonic stem cells. Now we reach the end of the webinar, and I'd like to thank you for your attention. In case of any questions, feel free to contact us per email at webinars@de.gbo.com. Take care and goodbye.